we are going to talk about gram-positive spore-forming bacteria. We will go straight into it. Gram-positive bacteria are bacteria which are categorized according to the color they turn in the staining process. The staining method uses crystal violet dye, which is retained by the thick peptidoglycan cell wall found in gram-positive organisms. The gram-positive spore-forming bacilli are the Bacillus and Clostridium species. These bacilli are ubiquitous and can live for several years in an environment because of the type of spores. Bacillus species are aerobes, while Clostridia are anaerobes. The members of this genus are closely related but different both in terms of pathogenesis and phenotypically. Their natural habitat is the soil or intestinal tract of the animals and humans. It is highly resistant to heat, dehydration, radiations, and chemical, and it appears as spherical, oval, or elongated in shape. The endospore for Clostridia is bulged, meanwhile the bacillus is narrow. The positions of endospore may be central, terminal, or subterminal, and the transcription and translation of the metabolic activity are absent. Gram-positive spore-forming bacteria were also not stained readily with dyes because of spore coats, and has DNA repair enzymes, and also destroyed at more than 100 degrees Celsius by burning and autoclave. The example of the bacteria are Bacillus anthrax, which bacilli have characteristic square ends, bamboo stick appearance, surrounded by polypeptide capsule. Spore do not stain by ordinary stain, and the lab diagnosis include gram stain, culture, PCR, and also direct fluorescent antibody tests. And the disease includes cutaneous anthrax and also intestinal anthrax. And the Bacillus cereus, we have fruit shape with square ends, straight or slightly curved, and non-capsulated, contain central spore, have petrotricious flagella, emetic and diarrheal. Lab diagnosis by serodiagnosis, direct detection method, culture. And the Clostridia botulinum have a rod shape, subterminal endospore, non-capsule, obligate and aerobe, and the diseases include foot botulism, wound botulism, and infant botulism. And for the Clostridium perfringens, it has large rectangular bacilli with rounded, truncated end, and pleomorphic with straight or curved roots, capsulated, central or subterminal spores, pathogenic, cause food poisoning. Lab diagnosis by ELISA. Effect on the food products. First, we have in the canned food, as canned food are processed under high temperature, C. botulinum, which is resists heat during the treatment, are more likely to cause spoilage. And the can swell and burst due to the gas production, cause partial digesting, putrid odor, increased pH, and productions of indole, carbon dioxide, and hydrogen. Canned product affected is corn and spinach. C. botulinum and bacillus can cause off-flavor, odor, and discolorations to the vacuum-packed chilled meat. Contaminations of meat are by C. perfringens on the soils and tanks and cause aggregations of creamy layer. Why are spores so difficult to control in food? This is because the spore of B series is active to different surfaces. The three characteristics that cause additions are high hydrophobicity, low spore surface charge, and spore morphology. Thank you!